So when we bring material in from the field, the harvested ears are wrapped in the individual pollination bag from when they were pollinated in the field. And each pollination bag has the date or the day of pollination written on the bag. So when the material comes in for processing, we can arrange that material according to which day it was pollinated. And generally we'll see some variation on the early days and the end days. And so those are the days that we want to give a little extra attention to. And you can kind of see a normal distribution in harvested days. So there's only one harvested ear from the first day, but we start to see more pollinations in the middle, and that's what we would hope to see. So then after the material has been laid out according to pollination day, our maize technician will do what we call a description of the material. She'll look at the kernel colors, the cob color, the kernel types, and the kernel arrangement, as well as some other characteristics, and log those into our database. After we have logged the information, we will pick the best ears for imaging purposes. So here we have the best 25 years, the most representative 25 years of this regeneration. And then we have a few leftover ears. This was a small regeneration, so there aren't a lot of leftovers, but we still have a lot of good kernels on here. They just don't need to be imaged for scans that go onto our website, and this gives us the best picture of what the material is and what we want to look for in the future. For maize populations, it's really important that we try not to change the gene frequencies from one regeneration to the next. And in order to facilitate that, we sample one kernel from every ear in that population to create a balanced sample. So each balanced sample will have a single kernel from each ear, and we will create 16 separate balanced samples. And generally we will keep 8 of those here in Ames, and we will send 8 balance samples to Fort Collins. When we're creating a balance sample, we want to shell material from the middle of the ear. These are the kernels that are least likely to have pollen contamination, and they're generally the best quality kernels. So we put one kernel in each slot. So when you have 100+ ears of a population, this can take a really long time. And then the sampled ears goes into the next bin. Once we have a kernel in each slot, we can slide that into the packet and take the next ear. For populations, we generally plant 400 seeds, aiming for 250 to 300 germinated plants in the field. And then in that number of plants in the field, we will typically make 200 sib pollinations with the goal of getting between 100 and 150 pollinated ears. Because the number of ears will vary from population to population, we will then in the next cycle choose either 2 or 4 full balance samples for that regeneration to ensure that the same number of kernels from each harvested ear are represented in the future population.